So thank you for that. I'm an adult psychiatrist at the University of Pennsylvania. I also work as a researcher at CHOP in neurodevelopmental illness. And I'm going to be presenting our technique for assaying synaptic, specifically presynaptic function in human-derived neurons, and some of the work we've done applying that to study the pathophysiology of STXBP1 haploinsufficiency. I have no conflicts of interest, but this work was funded by a generous gift from the Jerome S. Glaser Foundation, and that allowed us to expand the technique into the STXBP1 realm. So thank you for that and for the organizers for adding me late to this program. I'm going to be talking about relevant presynaptic mechanisms of glutamate release, specifically the ones we're able to assay and measure through our technique. Then we're also going to talk about how we apply that quantification to release properties in human-derived neurons, both potentially from iPSCs and ES-derived cells. We're going to talk a little bit about the core features of glutamate release and presynaptic mechanism in these cultures for the sake of giving a context to some of what we observe in the STXBP1 lines. In this case, an isogenic pair of the C663 variant that was generated at the CHOP iPSC core. So just as a brief refresher on presynaptic physiology, especially in glutamatergic terminals, presynaptic structures are defined by the presence of presynaptic vesicles, which are bound to specialized docking sites at the presynaptic terminal. These vesicles that are docked at docking sites are collectively known as the readily releasable pool. They're vesicles that would be fusion competent should the neuron fire and allow calcium into the presynaptic terminal. The vesicles that are not docked are often referred to as the reserve pool, and these are essentially the backup vesicles that will replenish the readily releasable pool once the releasable vesicles have moved. In the event of a depolarization and calcium influx into the presynaptic terminal, some of the vesicles will fuse to the plasma membrane. We can quantify this as a probability of fusion, which is essentially the number of vesicles that actually fused over the number that were fusion competent at the time of the depolarization. You could also quantify this as a probability of success. Was there any glutamate released at all? So in this system, we would see that the probability of fusion is 4/8. The success is 1 because there was glutamate released. Both these parameters are important because from them you can mathematically extrapolate how many release sites there are likely to be. You can imagine this, that if you have a high probability of fusion and you frequently observe the absence of glutamate release, that would tell you that there are very few terminals present. If you know that there's a very, very low probability of release, but you are actually frequently observing an absence of glutamate release, that would also tell you there must be very few release sites present. And we use this as part of our functional quantification. Once the vesicles have fused, they start to be replenished by those of the reserve pool. The vesicles docking is not an instantaneous process. It involves mobilization from the reserve pool and a complex docking mechanism. And the old vesicles recycle into what's known as the cycling pool, where they begin to reacidify, load with glutamate, and become fusion competent again. An important thing to note is that because the probability of fusion is always calculated relative to the number of fusion-competent vesicles, the absolute amount of glutamate can actually decrease even though the probability of fusion is stable. And this has to do with if there is a change in the number of fusion-competent vesicles in your terminal. So there are different measurements between probability of fusion and absolute glutamate release. The role of STXBP1 is thought to be facilitation of docking and stabilization, as we've seen in many of the talks here. So this raises the question, could haploinsufficiency impair fusion, docking rate, maintenance of readily releasable pool, and circuit synchrony? And we're going to look at all of these. We do this in a human iPSC-based neuron culture that has been enriched for mature synaptic properties. We do this with a neurogenin-2 overexpression system. Combined with fluorescent biosensors. Our differentiation protocol is very standard to most of them, except for we maintain FBS in our differentiation at an elevated level of 10% longer than most typical protocols, and we image these cells around day 85. This allows us to look at synapse-rich cultures of human neurons plated on rat astrocytes. You can see here our rat astrocytes supporting our human neurons with a very high density of presynaptic terminals here labeled by synapsin. So once we have the ability to make synapse-rich cultures, we need to get fluorescent biosensors into these cultures so that we're able to do functional imaging. We do this using a piggyback transposase to integrate the various fluorescent sensors into the genome of our original line to produce biosensor sublines. We then differentiate those cells and mix them together in a final neuronal co-culture. This allows us to balance the ratio as well as how sparse the various sensors are. The two we're going to be talking about in this case allow us to measure both synaptic glutamate and synaptic vesicle cycling simultaneously. The synaptic glutamate is measured with a fluorescent probe called iGluSniffer3. This is a fluorescent green probe that hangs on the outside of the cell, and it is dim in the absence of glutamate, becomes bright when glutamate is present. So this allows us to directly measure release of glutamate into individual synaptic clefts. We also have made a novel humanized redshifted probe that is targeted to the lumen of synaptic vesicles. This is pH sensitive, being dim when acidic and getting brighter with alkalinization. As vesicles fuse to the plasma membrane, it raises the fluorescence level, and so we can track the cycling of vesicles in individual presynaptic terminals. These are spectrally distinct from each other, so we can produce maps of both the synaptic glutamate and synaptic vesicle signal, and we can do this over patterns of imposed stimulation or spontaneous activity to create synaptic activity maps. As I said, we often impose stimulation on these cells. We can do this with trains or single action potentials. In the case on the left, what you can see is the glue sniffer on top, the red vesicle probe on the bottom. With a train, you get a signature vesicle transient, which rapidly rises to a peak and then decays upon cessation of stimulation. With individual action potentials indicated by the small black ticks, you cannot measure vesicle dynamics, but you do have appropriate sensitivity to measure glutamate release in individual synapses. Using the glue sniffer, we can monitor spontaneous activity in these cultures, which will cease if you block the postsynaptic glutamate receptors. This effectively electrically isolates all the individual cells. At that point, you can impose stimulation with a field depolarization, shown by the black ticks in that section, and this causes evoked glutamate release into the terminal. These are actually action potential mediated events, even though it is a field depolarization, and they are blocked in the presence of tetrodotoxin, which would stop action potential conduction. And this is what the data actually look like. The left panel is a raw image. The right panel is a variance projection, and this to show you how spatially restricted these signals are. And you can see there is spontaneous activity with individual vesicles being released, and then when we impose a stimulation, there is a coordinated synchronized release of vesicles. In this case, we've essentially labeled a postsynaptic cell, and you are seeing many, many cells synapsing onto it, and you're seeing the glutamate released into those synapses every time we depolarize the culture. From this, we can actually quantify our live glutamate imaging, and this is essential because we ultimately want to be able to quantify release at individual synapses and then pull that to quantify across a cover slip, across a line, and then even pulling lines to say what is the difference between genotypes. For this, you need an absolute quantification of glutamate release, and we do this by exploiting the fact that glutamate release is both quantal and stochastic. So by varying the extracellular calcium concentration in the bath, which is the top numbers, and then imposing stimulation, you can see that you get higher levels of glutamate release at an individual synapse in 4 millimolar calcium, gradually decreasing as you get down to 0.5. In 1.1 and 0.5 millimolar calcium, you also observe failure of synaptic release, where your probability of fusion is actually so low that sometimes you don't release glutamate from an individual synapse. This allows us to construct plots of the average amplitude as well as the variance in amplitude at each of these calcium concentrations. And from this, we can use a technique called multiple probability fluctuation analysis to predict what the theoretical maximum release from an individual synapse would be. This works by creating this parabolic curve fit. Because this is a stochastic process, if it was maximally high in amplitude but invariant, that would be a probability of 1, so the second x-axis intercept marked by n times q. And if it was invariant but there was no release, that would be at the origin. Your maximum variance is when you have 50% of your maximum amplitude. And this gives us an absolute quantification for every one of our individual synapses. And because we can do this for every synapse and every recording, minimum of 30 synapses per recording, sometimes up to 300, we can actually create distributions of synaptic properties. So instead of having to represent things as central tendency of mean or median, we can actually look at the full shape of a distribution of the synaptic properties. And this is very important because many synaptic properties are either very rare, so they occur in a Poisson distribution, they are multimodal, or they are bounded because they are probabilities, and therefore they do not follow Gaussian statistics. Here you see an example of the sensitivity of the probability of success to extracellular calcium, as well as the sensitivity of the probability of fusion. As you increase calcium, both of those parameters become more common, and so in this cumulative density plot, you can see that the curves tend to shift to the right. From those two values, we could then create a distribution of the number of synaptics, of the number of release sites at every synapse. There's an important thing to note about these particular cultures and something we've observed, and that's that synaptic depression is extremely common in these cultures. This has been described before, and even at stimulation frequencies, you can see this in the upper left, as low as once every 3 seconds, you can actually see depression of release in these terminals. This occurs particularly at high calcium and is evident until about 2 millimolar. If you use lower frequency of stimulation, you don't depress the terminal. We want to understand what is the mechanism of this, because the understanding the mechanism of depression allows you to interpret a lot of our subsequent data. So what we did is we measured these parameters in a depressed state, so the top panel of panel B, and then we said if we're measuring our properties in a depressed state, and then we mathematically extrapolate what the undepressed state would be, what model matches the empiric data of the undepressed state? And you could imagine that this is either due to, as I showed in my first panels, the number of functional release sites decreases due to a slow vesicle docking. So it's not that your probability of fusion goes down, it's the number of release sites go down. Or it's depression due to a decrease in probability of fusion. It turns out it's the first, and that matches the empiric data. This ends up being very important for subsequent interpretation. From this, we were able to build a model of vesicle dynamics and glutamate release for analysis of this data. We've done both empiric validation of the model, and from this we're able to actually extrapolate additional synaptic parameters from the ones we can directly measure, the most pertinent being that we can estimate the mean transit time of a vesicle. So how long does it take a vesicle to move from the reserve pool into being fully docked and fusion competent? So this brings us to STXBP1 and what the evoked phenotype is. In all of these slides, the control line is represented in cool colors. STXBP1 is in warm. You can see as you increase calcium, you get an increasing probability of fusion, but there is no significant difference in the probability of fusion between these two lines. This is also represented when you plot the probability of fusion as a function of calcium. What is different is in the middle panel here. The mean vesicle transit time measured at high calcium is significantly delayed in the STXBP1. So this would suggest that even though you have the same ability for a vesicle to fuse as long as it is docked, the time it takes for a vesicle to be docked and become stable is longer in the STXBP1 cells than the control neurons, with a median of around 8 seconds as opposed to about 5 seconds. We also see a trend towards a decrease in release site density. This is preliminary data with a relatively small sample size of 6 cover slips, about 800 synapses per group. And so we can't quite resolve this yet, but the trend is heading towards it being fewer release sites in the STXBP1. If we pair this with our vesicle imaging, we can get additional parameters. We can look at things like the endocytosis recovery time constant. We can also look at quantification of how large the readily releasable pool would be. We do this by imposing high frequency stimulation and high calcium when we have blocked functional endocytosis and vesicle replenishment, so you've removed that part of the signal, and that's what the second half of the graph shows. And from this, we can estimate things like the total pool of cycling vesicles, the readily releasable pool, and again, a measure of docking kinetics. This is very preliminary data, again, actually from only 1 cover slip per group, but the readily releasable pool appears decreased in STXBP1, as does the slope of vesicle docking, indicating the docking is likely delayed. And these data are summarized here. Again, the main takeaway, the fusion probability is not necessarily changed, but the speed of vesicle docking and stabilization is. We can also use these data to look at spontaneous activity. And this would be if you don't block anything in the culture and you just allow the cells to do what they do, you see a lot more spontaneous and erratic activity. We can measure this across both of our genotypes, and again, taking multiple synapses all together, we can create coactivity maps of synchronized release. This allows us to segregate the data into coactivity thresholds, so how synchronized is the culture. And because we have both amplitude, timing, and synchronicity data, we can then analyze differences either per synchronized event or per synapse. We do this by looking at the distribution of coactivity of these parameters under different coactivity thresholds at different calcium levels. We then take the area under the curve to have a single value per synapse. Most pertinent takeaway here is that the STXBP1 has a decrease in the number of synapses that participate in synchronized events. They also have a decrease in the effective signal to noise of signaling, meaning their unsynchronized release is actually higher in amplitude relative to synchronized release. The timing is also significantly skewed. In this case, this is a kernel density estimate of timing interval. You can see the STXBP1 skews to the right, indicating that there is a longer raw interval between events, and that distribution tends to be multimodal. This occurs across all calcium levels until you get to 4 millimolar, and even if you normalize to the mode of the activity to remove the raw timing element, these changes still persist, that the STXBP1 is more variable, multimodal, and erratic. And this can be quantified across multiple parameters describing both of these distributions. And so, again, the main takeaways of everything here, there appears to be a functional decrease in readily releasable pool. There's a maintenance of fusion properties, but there's delayed vesicle docking and stabilization, and this likely contributes to asynchronous activity and a reduced signal-to-noise ratio in the ability to encode information through synchronized circuit activity versus unsynchronized activity. And we're working to adapt this to a high-density method, hopefully for therapeutic testing, as well as screening of genotypes across multiple neurodevelopmental disorders, and scaling it up to be able to compare across in not only isogenic allelic series, but also individual patient lines, and then clinically defined groups. So thank you. This work was conducted in the lab of Stuart Anderson, again, funded by the Jerome S. Glaser Foundation, and taking questions.
Adam, thanks. I loved that. I wanted to ask actually about the maturation protocol. You guys, it looks like you take your eye neurons out to day 85, co-culture with rat astrocytes, right? This is considerably longer than a lot of other groups in the literature that you see. And so I'm wondering what drove that decision making, and are there particular markers of synaptic maturation in function or morphology that you think need to be hit to reveal some key phenotypes?
So our criteria for that is that the glutamate signals need to be spatially restricted and quantal. Until, at least by measurement with the glue sniffer probe, until you get to about day 65, although there is electrically evoked glutamate release and the cells are electrically excitable, the release does not follow quantal properties. It also does not appear nearly as spatially restricted until that point. In the absence of rat astrocytes, we actually do not observe glutamate becoming truly quantal. So that was the main criteria for taking it out that long.
Thanks.
Hello, that was a great talk. Related question about the requirement or not of the 10% FBS throughout the entirety of the culture. Is just to keep the cells attached till 85, or you find that there are components in serum that promote the maturation and the differences you're finding?
I'm not entirely sure of the answer to that. It does appear to help the astrocytes remain healthy longer. One of the issues is that we need to use mitotic inhibitors early in the culture to stop the rat astrocytes from dividing. So over very long periods like this, we have, a mitotically arrested astrocyte culture, and I think that elevated serum helps maintain their health and their adherence. And the presence of the astrocytes certainly helps with synaptic maturation, but it also helps with imaging, because when the astrocytes are present, you tend to get very diverse networks of neurites that are spatially defined from each other. If things are too dense or they cluster, it becomes impossible to do the imaging, either because you can't see a sufficient number of distinct synapses, or they're so close together that you can't resolve quantal properties, because essentially it starts to look continuous because you have so many superimposed on each other. So there's also a matter of balancing the density of your signal.
Thank you.
Very short question. Great talk. Thanks so much. A question about temperature. At which temperature did you do this measurement?
37 degrees.
Okay.
Always grown at 37 degrees and done with active perfusion with oxygenation at 37 degrees during imaging.
Oh, thanks. And what is your take on the majority of your effects you see at 4 millimolar calcium, right, which is high?
Yeah.
What would be the underlying mechanism for this?
So I think in the evoked data, the 4 millimolar calcium is elevating the probability of fusion to the point that even with modest stimulation protocols, you're actually depleting the readily releasable pool and not allowing it to replenish. So that gives you a way of resolving certain aspects of the biology that you wouldn't resolve otherwise. It's not meant to be physiological. It's sort of meant to show what can the system potentially do. When we do the spontaneous imaging and we raise the extracellular calcium, we're dealing with a culture that has no input or seeming function. It's just a network of excitatory neurons in a dish, and we're trying to mimic what it would be like if you had a higher degree of input driving the culture, causing elevated synchrony and elevated activity. In both cases, I wouldn't interpret it necessarily as being because of elevated calcium. It's meant to represent what those conditions would look like. And I think it's somewhat expected that in the conditions where you have higher activity and you are more likely to deplete vesicles, you start to see things like asynchrony of the culture as the individual terminals actually run out of vesicles at different rates, and due to the delayed replenishment, they remain out of sync with each other. So again, I would say that this is more about trying to model different scenarios that could exist in an actual developing neuronal circuit than it is saying that 2 or 4 millimolar calcium would ever be the actual way that that occurs.